Good morning, everyone.
Good morning.
How's everything going?
Not too bad.
All right, let's see who all we have here. All right. Um, I'm still working on protocols for you. I'm not sure if Nathan has finished his yet, but we're going to get those posted on the Moodle site as soon as we have them done. I went out into the field yesterday to basically make some videos showing you how to do some things in the field. I still have to edit those to get them posted. So anyway, as soon as we have this stuff up, we'll let you know. The equipment that you'll need to use is going to be in the lab, and we'll let you know when we have the protocols posted. We'll put in the protocol where the equipment is. Most of the equipment that we'll be using is really simple. It's things like an insect net and kill jars and things like this. So there's nothing terribly complicated about any of the things that we're going to be doing. So once you get the protocols that apply to your group, if you want Dr. Judd or I to go out with you when you go out to collect for your first time, set up an appointment with us and we'll be happy to do that. In some cases, the collecting you can do on your own so that, you know, the goal of each team is to collect a certain amount of whatever it is that needs collecting. Let's say that you're in the pollen load group. You have a certain number of insects that you need to collect of a certain type, and you can do that on your own. In some cases, it might be a little easier to do it with somebody else, but it's not impossible to do. There are going to be other cases, though. For example, with the pollen load group, you're going to need to measure the density of plants in the area and to make counts of pollinators in that area in order to try and get an estimate of density of pollinators of different types. That's going to require laying out a rectangular grid, and that is going to be easier to do with your entire team in tow. But that's the only part of that particular segment of the project that needs an entire team. And so I would probably schedule that, like, okay, today we're going to go out as a group and we're going to, you know, knock out these quadrats that we have to do, and then that's it. So that said, eventually you're going to have data, and you guys are probably going to have a lot of data when it's all said and done. And so we usually pause at around this time, usually it's a couple of weeks later into the semester, to talk to you about data analysis. And in lecture, we talk to you about data analysis in terms of, you know, what are these people in this paper that you're reading doing, so that you can understand, at least in concept, what the people are doing. But when it comes to lab, though, you're actually going to have to do the analysis yourself, and we'll help you with it, but we want to start introducing you to the ideas. How many of you watched the screencast that I asked you to watch overnight? Okay, less than half. Okay, so maybe I should go back and reiterate that. Those of you who did watch it, was it clear? Were you able to follow, or what? Are you still confused by it?
I could follow some of it, but there's still a little gray in some areas.
Okay, what about the rest of you? Nodding of
Yeah,
agreement from McGilla. Tessa, you're still struggling. Okay, then maybe what we should do is we should just start there. So let's see. All right, I'm going to begin with analysis of variance because that's kind of the most straightforward thing. We're actually going to have you in class work through an example of the analysis of variance. And so you're going to see this again, but it's a good idea to have it in your mind as you approach data collection. It's always good to know what you're going to be doing analysis-wise and to have that in your mind. All right. Share screen. All right, can everybody see the analysis of variance slides?
Yes.
Okay, all right. For some reason, I can no longer see any of you. Why is that the case? I love Zoom. No, I do not. All right. So basically, this is part of the slideshow that we'll show you later on in the semester. But the beginning of the slideshow is the part that I want you guys to pay attention to today. So in this case, this is actually just an example from a textbook. In this example, they did the weirdest of all experiments that I've ever seen, which is they watered pea plants with different concentrations of different sugars. So the control group got water, just plain old water. Then another group got a 2% glucose solution, a 2% fructose solution, a 1% glucose, 1% fructose solution, and a 2% sucrose solution. I guess my kind of when I project onto why somebody would do this, it's the idea that plants need sugars in order to get energy. When they do photosynthesis, they produce glucose. And so if you gave them glucose in their water, would they do better or something like that? And so they're putting in a 2% solution, but in one case it's glucose as a simple sugar, one case it's fructose as a simple sugar. In one case, it's the combination of both of those simple sugars, but they reduce the concentration, 1% glucose, 1% fructose, so that it all adds up to 2%. And then in the 2% sucrose, sucrose is just a disaccharide that's made up of glucose and fructose. So that's I think, the logic of it. Whatever the logic of it, it doesn't really matter. We're just using it as a data set to play around with numerically. What they did was they then measured how much the pea plants grew afterwards. And so when you look at this thing, you can actually just plot the data. And so we have the various treatments as you go up the y-axis, and we have the growth increment on the x-axis. And you can see that each of these treatments resulted in some cases, different results. The control is very variable, but also seems to have a higher mean than the rest of the groups. The 2% sucrose has a lower variance than the control, but is about the same in terms of its variance with the other ones. But also it looks like it has a mean that is intermediate between the control and these other three. And these other three don't really look very different from one another if you just plot the data. So we could actually calculate the sample means and the 95% confidence intervals. And I've done that a long time ago, many years ago, and I've basically plotted them on here. And you can see that when you look at a mean and a confidence interval, remember that when you're trying to determine graphically if two means are different, you ask yourself, does this mean belong in this means confidence interval? This mean lies outside of that confidence interval. This mean lies outside of that confidence interval. Therefore, we would probably conclude that these two means are different. Remember, and some of you made this mistake on the exam, it isn't about the overlap of the confidence intervals. It's about whether or not the confidence interval includes another sample mean. And we made that point in class, and it didn't register on some of you. Here, it looks like these means are all part of one another's confidence intervals. If you draw lines vertically from the x-axis to where the confidence intervals are, looks like these things are not terribly different from one another. This is different from all of these, and this is different from the control, and the control is different from all of the other treatments. So when you have multiple treatments like this, one of the tendencies is to just fall back on things that you already know. For whatever reason, in high school nowadays, they teach people the t-test. And so one of the questions that you could ask is, why not just do lots of different t-tests? Well, the problem is that the more comparisons you make, the more likely you are to make a type 1 error. And we haven't talked about type 1 error yet in class, but a type 1 error is when you conclude, and in this case, because we're comparing means, we're going to use the language of means. You conclude that two means are different when they are not. And so this is a type of error. It's a decision error. And because you're making statistical estimates of things, there's always a probability that you're going to make the wrong decision at the end of something. And so a type 1 error is the kind of error that you don't want to make too terribly often. And so we usually set the type 1 error rate, which is alpha, at 0.05. That basically is a 5% error rate. So if you do 100 statistical tests in your lifetime, and you're always testing at the 0.05 level, at the 0.05 level, chances are that 5 of those decisions were wrong. You said 5 times out of 100 that two means were different, when in actuality they're not. And this is actually where the 95% confidence interval comes from. If we set our alpha at 0.1, which is making this kind of error 10% of the time, our confidence intervals that we would calculate would be 90% confidence intervals. And so the confidence interval is the reflection of the alpha level. So alpha level is the type 1 error rate. And so we set that at the beginning. And it's up to the researcher as to what that error rate is. If you're an ecologist, maybe a 0.05 level is too stringent. Maybe you want to relax your type 1 error rate because the consequences of occasionally making a wrong decision about two means being different isn't that big of a deal. But a cancer researcher who has a chemotherapeutic agent that might cure a really bad case, a really bad type of cancer like glioblastoma, which is fatal in like 6 months after you're diagnosed, and you think that your drug works, but in the process it also kills your liver because it's so toxic, you want to make very sure that that drug actually gets rid of the cancer. And so you might not think that a 5% type 1 error rate is sufficient. You might want that to be a 1% type 1 error rate. If you say that the placebo is different from the cancer drug, you really want to make sure that that difference is a real difference. So the problem with type 1 error rates is that when you do lots of multiple t-tests, each one of these comparisons has a type 1 error rate associated with it. So if you compare control to sucrose, that has a type 1 error rate. Compare control to glucose, it has a type 1 error rate, et cetera, et cetera. And to do all possible combinations for this data set that we just looked at requires 10 tests. So the type 1 error rate for those two tests would be 10 times this, which then makes it a type 1 error rate of 0.5. And so your type 1 error rate is now not 5%, but it's 50%. And that is basically the equivalent of flipping a coin. You might as well just not do any experimentation and flip coins to determine whether or not two means are different. And we really don't want that to be the case. So what we're going to do is we're going to employ a method that basically allows us to compare all of the means simultaneously without inflating our type 1 error rate. Are you guys clear on type 1 error rate and what alpha is? Okay. So basically, analysis of variance is called analysis of variance because it's all about variation. So here's an important distinction for you. Analysis of variance really is analysis of variance. And you might say to yourself, well, but Dr. Klowitzki, we're using analysis of variance to distinguish between means. Why is it not an analysis of means? Well, because it's an analysis of variation, but there are different kinds of variation. There's variation that occurs among the means, and that's how we actually determine whether means are different. But our view of whether or not those means are different can be obscured by variation within, and then there's the total amount of variation. And so what we're interested in analysis of variance is how much variation is there among the means versus how much variation there is within the means. If the within sample variation is really large, it will obscure any differences among the means. And so you'll also see that this is SF. SS stands for the sums of squares. Now you'll remember that when we calculate sample variance, what's in the numerator of the sample variance equation? Anybody? Just one question. Don't forget it because it comes up over and over again. It's about to come up again. Joseph? I can't hear you. Are you talking, Joseph? I still can't hear Joseph. Can anybody else be heard? You guys can hear me, right?
Yes, it's the number of samples minus 1.
That's in the denominator. What's in the numerator? Joseph, unmute yourself if you want to speak.
Okay, can you hear me now?
I can hear you now.
Cool. So it's like the value, like the observation value minus the mean, and then that quantity squared.
And then you add that up across all of the data points that you have. Yeah.
Yeah.
So when we say the sums of squares, we're talking about the sums of the squared deviations from the mean. So sums of squares is really a shorthand notation. It's the sum of the squared deviation from the mean. What's the value? How far is it from the mean? Square that, add that up across all of the observations, and then divide it by that. And so this numerator of the sample variance is the sums of squares. So when we went through calculating variance, I told you, remember this equation, because of all the equations that you should remember this one, and in particular the numerator of this one, because when you see SS among, that's a sums of squares. It's a sums of squared deviations. Now, because we're talking about it's the sum squares among the treatments, in this case, we look at the treatment means minus the grand mean. and we square that up across all of the different treatments. That's what sums of squares among is. And we're using the sample mean minus the grand mean because it's among treatment variation that we're trying to quantify. Sums of squares within is just this. It's the individual values minus the mean summed across all of those values in that sample, in that treatment, but then also summed across all of the different treatments. So you do it within each treatment, but then you also do that across all the different treatments. And so these two types of variance are basically just based on the same ideas that a simple sample variance is based on. So in a way, the analysis of variance really isn't anything new conceptually. It's something that builds on something that you already know about. So one way of thinking about this is here's the grand mean. Sums of squares among basically says, what's the difference between this sample mean and the grand mean? Square that, save it. Difference between this sample mean and the grand mean, square that, save it. This mean to the grand mean, square it, save it. This mean to the grand mean, square it. This mean to the grand mean, square it. Add all those up, that's sums of squares among. Now, sums of squares within is how does this value differ from its mean plus this value squared, this one from this mean squared, plus this to that squared, this to that squared, this to that squared, this to that, 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 all squared within this sample, plus this minus its mean squared, this minus its mean squared, this minus this mean squared, this to that, 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 all squared and added up. Same thing for this sample, same thing for that sample, same thing for that sample. So it's the individual values minus the mean squared for all the members of that treatment, but then added across all of the treatments. So summed within a treatment and then summed across treatments. So sums of square total can be calculated separately if you want. Oftentimes we just take sums of squares among and sums of squares within and add those two things together, but we can also calculate it independently and check our math if we wanted to. So sum squares total is basically using the grand mean, and it asks, how is this value different from the grand mean? Squares that, saves it. This to that squared, this minus 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 that squared, then this minus that squared, this minus that squared, the green minus x squared, sorry, the mean minus the grand mean squared, this minus that, once again across all of the different treatments. So it's basically measuring how does each value differ from the grand mean. And so if we were going to write sum squares total up here, it would now be each individual value minus the grand mean squared, summed across all treatments and summed within all treatments and then summed across all treatments again. So you can calculate it independently if you want. So what does this look like? We have the sums of squares among that we've calculated by looking at how each mean varies from the grand mean. We've looked at sums of squares within, which is a measure of how sample variation within samples is. And then we basically divide each of those by the degrees of freedom. Now remember that when we calculate sample variance, all we did was we took the sums of squares, the sums of the squared deviations from the mean, divided by the number of degrees of freedom for sample variance. And so once again, this MS, which is the mean squared, is nothing new to you. It's just another derivation, another variation on variance. So it's just the sums of squares divided by the number of degrees of freedom for whatever this is. The degrees of freedom for the among number of degrees of freedom is the number of treatments minus 1. So in this case, we have 5 treatments, so we have 4 degrees of freedom. And in this case, the sum squares within, because we're doing it across all of the treatments, we add up the sample sizes of each of the treatment and we subtract from that the number of treatments. So in this case, we have 5 different treatments. Each treatment has 10 observations in it. We have 50 observations total. We have 5 treatments total, so we would have 45 degrees of freedom here. 45 degrees of freedom minus 4 degrees of freedom. I'm sorry, 45 degrees of freedom plus 4 degrees of freedom is 49 degrees of freedom, and 49 degrees of freedom should be the total degrees of freedom, which is the sum of all of the sample sizes of all the treatments, which is 50 minus 1. And so once again, the math should match up within the table. And then the F statistic is just a measure of how variable are the means from the grand mean versus how variable are the samples within themselves. So it's a ratio of variation among samples divided by variation within samples. And the F statistic is actually what we evaluate to make the decision. One way of thinking about this here we have sample mean 1, and here we have sample mean 2. And if we're interested in knowing whether these means are different from one another, I mean, they are different from one another in that they're actually numerically different values. But the question is, can we distinguish whether this mean came from the same population as this mean and vice versa? We have to know something about their variability. So let's say that the samples that these things came from are highly variable. Such that when we go to calculate their 95% confidence intervals, the 95% confidence interval for mean 1 is really wide, and the confidence interval for mean 2 is really wide, such that this falls within the confidence interval of that one, and this falls within the confidence interval of this one. We would conclude that they are not different, because this mean could have easily come from the purple population, and the purple mean could have easily come from the brown population. But now imagine that these two means are exactly the same distance apart in terms of their numerical value. But now, instead of these having a lot of variability in the sample, these samples are really not variable at all, not very variable at all. Now, the 95% confidence intervals are relatively narrow. And now we would say that these two means are different from one another simply because this mean could not have come very easily from the brown population, and the brown mean could not have come very easily from the purple population. Nothing's changed about the difference between the means. That value is the same. The grand mean is somewhere here in the middle. The distance between this mean and the grand mean is the same as the distance from this mean and the grand mean as they were before. What has changed is the variation within the samples. The more variation there is within the samples, the harder it is to see the differences between the samples. The harder it is to see the differences between the means. So it's a ratio of how much variation is there among the means divided by how much variation there is within the means. As variation among the means gets big and variation within the means gets small, F gets big, and then when F gets big, you have more likelihood of finding differences. If there's a lot of variation within the samples and not much variation among the samples, F gets small, and small F statistics are associated with things not being different. Okay? Does that make sense? Intuitive sense too? Okay. So we can work out this example. So this is just the calculations. The mean, 70.1, 59.3, 58.2, 58, 64. It looks like those means are probably different from one another. They're all sample size 10. We calculate sums of squares within by subtracting 70.1 from 75, squaring it, 67 minus 70.1, squaring it, 70 minus 70.1, squaring it, 75 minus 70.1, squaring it. Do that to all of those, add them up. That's the sum of squares within. Do that for every treatment that you have and add all of that up, and it should equal 245. That's how you calculate sum of squares within. It's just like calculating the sample variance, but then you just add those sample variances, the numerator of sample variance, and you just add those up across all of the different values. Now the grand mean is here at 61.94. If you added all of these values up and divided by 50, it would give you 61.94. Also, if you added these 5 values up and divided by 5, it would also give you 61.94, but only because each of these is based on the same sample size. So normally you would just add the whole data set up and divide by the total sample size of your data set to get that grand mean. But sum squares among is the sample mean minus the grand mean squared, and then you weight it times its sample size. So sum of squares among these values are different. They should all be multiplied times 10. So this should be 665.9. This should be 69.7, 139, 152, et cetera, et cetera. And if you do all of that, it should add up to be 1,077. So the mean minus the grand mean squared times sample size, which is 10, is what that should equal. And then we have sum squares total, which is 1322, but that's simply 245.5 plus 1077.32. So this and this should add up to this. But you could calculate this independently by taking 75 minus the grand mean, squaring it, 67 minus the grand mean, squaring it, 70 minus the grand mean, squaring it, for all of the control, all of the glucose, all of the fructose, all the glucose, fructose, and all the sucrose. Add all of that up, add all of those squared deviations from the grand mean, and it should be 1322.82. So all that math should be internally consistent. And then it's just a matter of putting together the ANOVA table. You have 4 degrees of freedom for among treatment variation, because there are 5 treatments and it's 1 minus the number of treatments. 45 because you have 50 observations total and 5 different treatments, and that should equal 49, which is 1 minus the total sample size. Mean squared is 1077 divided by 4. Mean squared here is 245.5 divided by 45. Your F statistic is really big, which means your p-value is really small. Now, what is p? P is the observed type 1 error rate. So it's the type 1 error rate for your specific data set. So if you say that 2 means are different from one another, for this particular data set, it's way lower than 0.000345 6. It's way less than this. And so the chances of the chances of 2 of these means being different from one another is actually really high, because the chance of you saying that they're different and making a mistake when they're not by saying that they're different when they're not different is really, really low. What we do is we compare P to alpha, and we ask ourselves, is the P that we observe lower than the alpha level? And so if this is the type 1 error rate that we are finding acceptable, we can make as many as 5 type 1 errors per every 100 tests that we run. The question is P smaller than alpha? And if P is smaller than alpha, then we conclude something in there is different. At least 2 of the means differ from one another. And we've already kind of talked about which differences are there. If P was greater than alpha, this is how we conclude that things are not different. Because we could say that they're different, but if we said that they're different, if P was, for example, 0.1, we have a 10% type 1 error rate. 10% of the time we would say things are different when they're not, and that's higher than what we said at the beginning was acceptable to us, which is only 5% of the time making that type of error. So P tells us whether or not we've crossed that boundary for that criterion for saying things are different or not. And it's a calculation based on your data. So this F value at a certain number of degrees of freedom in the numerator and the denominator will give you this P value. And that's how you evaluate whether or not things are different. So when we look at this, we can go into R and we can create a figure that shows the means and their 95% confidence intervals. And it's clear that there are differences. Control is different from everything else. The sucrose is different from the control and also different from these 3 simple sugars, and all of the 3 simple sugars are not significantly different from one another. So conceptually, are you clear with how it's working? Okay, then let's stop sharing. Somehow I can stop sharing. So here on my, let's see, let's go and share again, actually now that I'm here. Can you guys see a frog? Okay, so I've taken the R scripts and the data sets that are on Moodle. So I posted some Moodle scripts for this week in lab. And I've put them all in one folder. And once again, if you set up your computer correctly, and if you click on this so that you can get info, you can set up your computer such that it will always open things in RStudio, and so you can make that a change that applies to every R script that you have, so that when you look at the P's analysis for Bio-133, which is an R script, when you double click on that, it will just automatically open that script up in R. I say it will open it up. It's slow today. It didn't get its coffee or something. Ah, there it is. So here's the script. So remember that R has these 4 panes. This pane here is the pane where you can edit your script. So this is the script, and this is what it looks like. This is the console. This is where your code is actually running. This is a history and environment section where it shows you the things that you've done. And then this is a place where you can see the plots that you've generated, and you can also load packages of different types. And you can see that right now there are relatively few packages loaded because I just opened it up. So I don't have checks next to any of these things, although I'm going to want to load some things. Now, in this particular script, there is a ton of commentary. So at the beginning, there's some general notes because I know that you guys are still new to R. It explains what you can do with hashtags, and hashtags you can make things such that you compress all of your stuff down. But hashtags, a hashtag before a line basically is a comment. It's never going to run. It's not actual scripting language. It's just notes to yourself. The first line of actual executable code comes on line 33. Get WD is just short for get working directory. I always check this at the beginning because I want to know that I'm working in the place that I think I'm working in. So down here in the console, I've basically said to the computer, where on my computer am I running R from? Well, I want to run it from where my data are, and my data are in Bio133 lab stats, which is this folder that I put on my laptop yesterday. So it's on my desktop, it's right here, it's in the beak of the Vultr, Bio133 lab stats, and I'm basically checking to see, am I working where I think I'm working from? A lot of times students don't have a good feel for what their directory structure is on their computer, and so they put R in one place, they put their data in another place, they put a script in a third place, and R doesn't run because R doesn't know where to go and get your data because you're running your script from one directory and your data resides in another directory. And directory is just old school way of saying folder. So you can actually set what working directory you want to have. You would do set WD, and in parentheses, you would give it the path to where that is. So this whole thing down here is the path to where your file is. If you want to have access to that path, so for example, my folder is on my desktop, and it's Bio133 lab stats, I can right click on that, and when I go to get info, the path to that folder is right here. It's in users Chlewinski P on my desktop and in the name of the folder. And you can see that the path here is users Chlewinski P desktop and the name of the folder. So you can put the path in there manually if you want. I just find it easier to open my script where it is, where I want it to be. So the first thing you need to do is read in the PEAS data set. So the PEAS data set is in a comma delimited file, a comma separated values file. And this is basically just saying read.csv. And in our language, let me explain what this script means to you. I like to have shorthand names for things. And this thing here, which is the less than sign and a dash, this is called the assignment operator. It actually has a name. The assignment operator basically says, take whatever is over here and call it that. So that I don't have to write what's over here over and over and over again. I can just write the shorthand name and it knows what I'm talking about. So in this case, what we want to do is we want to read in the CSV file. So that's read.csv. And then the file name that we want to read in. And the file name goes in quotes. So what we're basically saying is I don't want to write, every time I access this file, I don't want to write this whole thing out. So I'm just going to call it PEAS for short. So whenever I type in PEAS, it's going to run this line of code because I've assigned this line of code that shorthand name. So the assignment operator is your friend because it allows you to make shorthand notations that mean longer, more complicated things to write out. Okay, and you'll see when we get down to making graphs, this becomes really super useful. So the first thing you want to do is read in your data file. Now, you execute code on the Mac with command return, and on a PC, it's Control and Enter. And you can see down here that it seems to not have done anything, but whenever you run a line of code and then it gives you the command prompt right after that, it basically means you ran the code and it didn't have any problems, which is good. But you never trust computers. Don't ever trust a computer. Computers lie, they cheat, they steal. They cannot be trusted. So you always want to make sure, did it really do the thing that I think it did? So head basically means give me the first 6 lines, and give me the first 6 lines as P's. Well, what is P's? Well, P's is me reading in my data file. So I want to just look at the P's data file real quick to see if it looks like it read it incorrectly. And it looks like it read it incorrectly. I've got control, I've got the growth increment, I've got some other stuff that you don't really need to worry about. I've also put a thing here at the beginning of the data file called treatment A, which will help me order things later when I go to make graphs. I'm going to attach P's. This basically puts the P's data file in a slightly easier form, so we don't have to write out a lot more things. If you don't attach your data file, every time you refer to a variable, you have to say, I'm looking for the variable in the P's data frame, and I'm looking for variable growth. If you attach your file, all you have to do is tell R growth, because it knows that you're working with this data frame. Okay? So it just saves us some keystrokes if we attach it. Sometimes when you attach a file, the act of attaching the file doesn't work with certain packages, but it works for all the packages we're going to be using today, so it doesn't really matter. So the next thing you have to do is you have to load packages. We're going to need ggplot, we're going to need rmisc, and we're going to need car. These are just packages that do different kinds of things. It's going to give you some error messages, but these aren't errors. These are basically just telling you things. Hey, ggplot was built under a different version of R, so that you're aware that if things aren't working, maybe it's because the package was built under a different version of R than the version of R that you're running. And this is just saying, hey, I'm loading some packages that this package needs to run. And as you do this, you see that these packages now have checks in front of them. ggplot has a check in front of it. Lattice now has a check in front of it. We just saw that go by. rmisc up here should have a check behind. Oh, plier has a check by it. rmisc has a check by it because we've loaded all of these packages, and so now we're ready to do stuff. So I always check the class of my variables to see if they're what they're supposed to be. So the treatment is a factor, it's not a number, and so I can check that. Growth should be a number, and in this case, it's an integer number. We're going to skip these classes because those are for multiple comparisons, which we're not going to do today. So we won't worry about that. So now we're down here at line 141. We want to just check once again to see that the P's are still there. And then what we can do is we can do a thing called Shapiro test. So when we do an ANOVA, ANOVA has a lot of different assumptions. One of the assumptions is that the data are normally distributed. And the way we test that is with a thing called the Shapiro-Wilks test. And we can actually do this for each treatment by itself, not the recommended way of doing it, but it's one way of doing it. And so T-apply basically says, I want to apply to all the treatments looking at growth a Shapiro's test. And so that's kind of what the lingo is. And so it's doing a normality test on each of these different subsets of the data. And once again, the p-value should be higher than 0.05 because we want it to be not different from normal. And so the control is not different from normal. Fructose is not different from normal. Glucose is not different from normal. None of these are different from normal. That's probably because we only have 10 observations. So we satisfied that requirement. Go down to line 209 is the next useful code. The Levene's test, one of the other assumptions of an analysis of variance is that the variances of each treatment are similar to one another. And when we run Levene's test, we actually find that the variances are different. from one another. So we can get R to print out a summary of things. So summary SE is a function within R. And what we're doing basically is we're saying, I want summary data, means, standard errors, confidence intervals, etc., on the data file P's, but I don't want the entire data file P's. I just want the means of growth, and I want it divided up by treatment, and I want you to store that in a shorthand thing called CI plot. CI plot stands for confidence interval plot. This is how we're going to plot our data ultimately. And so once again, we can run that line of code. It doesn't give us any errors. And when we click on CI plot, what it has done for us is it has calculated the average growth of control, the standard deviation, the standard error, and the confidence interval. Don't you wish you had this on the last exam? So we have that for the 1-to-1 glucose fructose, the 2% fructose, 2% glucose, and 2% sucrose, and see how it has rearranged the order of the treatments in the output relative to what it was in the data file, because it does things alphanumerically. And so there are 10 observations of each. These are the means. And you can see that the control seems to be more variable than the other treatments. It's at a confidence interval of 2.85, and all the rest of these are down in the low ones. And that's the reason that the Levene's test has turned out to be significant. It's basically saying this variance, this standard deviation, is higher than the standard deviations of all of these other things. So normally we would do something about that. We would log transform it. But for now, I'm just going to not log transform it, because I want the graph that you get to see, that you get to look like the graph that you have in the PowerPoint. So this thing here, once again, I'm going to give a line of code, a shorthand thing called analysis, and AOV stands for analysis of variance. So basically what this line of code is saying is, I want you to do an analysis of variance. The response variable is growth. The treatment variable is treatment. The data is in the data frame P's, and I want you to store that result under this heading analysis. When I run that line, it just shows me that it ran without any problem, but it hasn't given me any output. If I want to see the output, I need to ask R to summarize the analysis. And when I do that, it's going to give me an ANOVA table. And you see that these numbers match up to the numbers that we calculated by hand. Sums of squares for treatment is 1077.3. Sum of squares for within treatment, which in R speak is residual variation, is 245.5. The F statistic is 49.37, and the probability, the observed p-value is 6.7 times 10 to the minus 16. So actually, I wrote it up here wrong earlier because I was misreading my own thing. It's 6.7 times 10 to the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This is the observed type 1 error rate for this data set. That is smaller than 0.05. So you can say with a high degree of confidence that at least two of those means are different from one another. You can actually figure out which of those is by doing a Tukey's honest significant difference test. This is a test that basically does all possible pairwise comparisons, once again controlling for alpha level across the whole experiment. And what you'll see is that any of these have a p-value below 0 are significant differences. And so we see that sucrose is different from the 1-to-1 glucose fructose. We see that control is different from that. We see that sucrose is different from fructose, control is different from fructose, sucrose is different from glucose, control is different from glucose, control is different from sucrose. So all of those differences that we saw when we looked at the figure are differences that we find when we actually analyze them using an appropriate analysis, the Tukey's on a significant difference test. So we can redo our CI plot where we have treatments as A, B, C, and D. This has them ordered the way they originally were, which was control, the simple sugars, then the compound sugar. But it's all the same. It's all the same means. They're just rearranged now. And I've done this because I want to ultimately make a graph. So why is that line there? That line is not supposed to be there. So what I've done here is all of the script between these two hashtags basically does something. So I've given this whole big line pplot. So because I've used the assignment operator, all of this code right here will be run when I type the word pplot in. So I'm going to run the pplot, and once again, no errors, but also no plot over here. So now what I have to do is I just need to call up that plot by saying pplot, and it will give us the graph. Hopefully. And there's the graph. There's the graph that we had before. Control, 2% glucose, 2% fructose, the 1-to-1 glucose fructose, and the 2% sucrose. And all of the differences that we found when we ran the Tukey's on a significant difference test are also apparent in the figure, because these are 95% confidence intervals. So let me just run through this script for you really quick. So I'm using a package called ggplot. It's just R's plotting package. I'm telling it to plot the data that are in CI plot. This is an aesthetics statement, and it's basically saying that I want you to put on the x-axis treatment A, and I want you to put the y-axis is going to be the growth. I want you to put error bars on this plot that are basically have a minimum and a maximum value. The minimum value is growth minus the width of the confidence interval. The maximum is growth plus the width of the confidence interval. And then this just tells you how wide to make the cap that is on the end of that line. I want you to not put a line connecting the dots with one another, which is why there's nothing in the parentheses. I want you to give me some points that are filled with white and ringed in black, which is what shape 21 is, and they're size 3. I want you to label the X labels treatment. I want you to label the Y axis growth increment. I want you to put a title on there that is seedling growth. And as a matter of fact, I can also put on this H just equals 0.5. And when I do this, except I need to be able to type correctly. When I do this, you can see that in the plot here, the title is not centered over the thing. It's left justified. That's strangely a weird thing that they've kind of added. When I put in HJust, I'm basically saying horizontally justify this, but put it in the middle. That's what the 0.5 is. So when I run this again, it now has the title. No, it didn't do it. Okay, I don't know why it didn't do that. Anyway, I'll figure out how to do that later. This is a theme statement, basically saying I want it to be a black and white plot. This is then me telling it what I want the order of the labels on the x-axis to be. So scale X discrete labels, I'm just telling it manually. Rather than putting an A and a B and a C and a D and an E, I want it to put control, 2% glucose, 2% fructose, etc. And then this is just a bunch of formatting. It's me telling it to get rid of the grids behind it, telling it what size to make all the fonts and things like this. So basically, it's just a bunch of formatting of how I want the graph to appear in terms of fonts and spacing and stuff like that. So that's the figure that comes out of this analysis, and it looks exactly like the figure that is in the PowerPoint that we just went through. Questions? Those of you who have to do an analysis of variance, we will help you adapt. So the nice thing about having a script that works is you can just adapt it to your data set. So when we get your data, if you have to analyze your data with an analysis of variance, we will just pull up the Pease analysis script. We will rename it with a different name, and then we'll go through and we'll start changing things in it. We'll change it to a new data file. We'll change the variable names, et cetera, but it will run through the same type of analysis because we have a structure that works. So why go back and rewrite all of this script from scratch when we can just adapt a script that we already have that works? Questions? You're not going to have to generate the script on your own. We will help you with data analysis, but I want you to have an idea of what's involved. All right. Do you guys need a little break before we start regression? Go to the bathroom? All right, take 5 minutes. I'll see you back here in 5. Okay, sorry, I got stopped in the hall while I went to the bathroom. Everybody back? All right. So regression. Regression is, strangely enough, exactly like ANOVA. It doesn't look like it, but the calculations are all super similar. And so actually, it ultimately also produces an ANOVA table from regression. So regression is when we're looking at the, hold on just a minute. I have to change all of this because I'm in Zoom land now. So regression is when we're trying to estimate the relationship between one variable and another variable. And usually we do this by relating those two variables with a linear equation. It's slightly different from correlation, where we're just trying to see if there is whether two variables covary, and we're not necessarily really concerned about the form of that covariation. And so it doesn't have to be linear. But people oftentimes use regression and correlation interchangeably. I try and be a little more careful with my language, although I don't always succeed. So we generally take the form y equals mx plus b, or y equals a plus bx. In this case, b is the slope value and a is the y-intercept. X is the x variable and y is the variable that you're trying to predict. And so when we do a regression, in theory, what we generally do is we have an x variable that we vary on purpose, and then we look to see how the y variable responds to that. This is what a model 1 regression would be like. The x variable is under control of the investigator, and then as we vary x, we look to see how y responds to that. And so oftentimes we will refer to an independent variable and a response variable, because that's in theory really what we're doing, is we're having independent control over the x variable, and then we're seeing how y responds to variations in x. The x's are fixed because we have control over them, and so once again, the beta is the slope, the alpha is the y-intercept, and mu is the response of the y variable. So there are some assumptions under regression, just like there are assumptions under analysis of variance. One is that the x's and y's are independent of one another, and that they are normally distributed around the prediction line. And then the other thing is that as you move along the regression line, the variation around the line doesn't change over the course of the range of x values. That's what homoscedastic means. Homoscedastic just means similar variances. Heteroscedastic means different variances. It's just fancy pants statistic talk for equal variances or unequal variances. Model 2 is a different type of regression, but it's where both x and y are random variables. Strangely enough, when most biologists do regression analysis, they actually have a model 2 regression, but they actually run ordinary least squares regression on the data, which is technically not what you're supposed to do. So I just want you to be aware of that. This is a situation where you don't have control over x, and you're once again looking at how x and y are related. So in this particular example, we have flower beetles. They're raised under different conditions of relative humidity, and the relative humidity in this case has been set by the investigator. They weighed beetles in 9 groups of 25 beetles, and they calculated the weight loss after 6 days of hanging out at these different relative humidities. So the independent variable in this case is relative humidity, because this is the variable that the investigator is controlling, and the investigator is looking for how does weight loss change in response to changes in relative humidity. And so weight loss is the dependent variable, or you can also call it the response variable. So these are what the data look like, going from 0 relative humidity to 93% relative humidity. And as you can see, the beetles seem to have lost the most weight at lower relative humidity and lost less and less weight as you got higher and higher humidity. So clearly these poor beetles are being dehydrated over time. We can make a graph of this. These are the data points as they vary, as weight loss varies with relative humidity. And we could actually ignore relative humidity and just look at the average weight loss. And the average weight loss is 6.022, I think, if I'm recalling correctly for this data set. And if we draw a line that is horizontal to the x-axis, parallel to the x-axis, this is basically the hypothesis of a slope of 0. And so the hypothesis that we're testing when we do a regression is the null hypothesis is the slope is equal to 0. The alternative hypothesis is that the slope is not equal to 0. It will either be a positive slope or it will be a negative slope. And so when we do a regression, we're testing the slope value, not the y-intercept. We're testing slope. Now, if we think of this horizontal line as the average, because it is the average weight loss, we could calculate residuals just like we would calculate residuals in an ANOVA. A residual is just a word for how far is this data point from the mean? How far is this data point from the mean? How far is this data point from the mean? So in ANOVA land, what we were doing earlier, this is a residual. How far is the x value from the mean? We square those residuals, add those residuals up, and divide by n minus 1 when we want to calculate sample variance, but we begin with residual variation. How far does each value vary from the mean? These residuals look like this. We know that this is not a very good fit of the line because these residuals out at low values of relative humidity are high in positive. The residuals over here are high in negative, which indicates that this line is not a very good fit. What regression analysis will do is it will figure out what is the best fit line through these points. And the best fit line is the line that reduces the overall size of the residuals, the overall sum of the residuals. So when we evaluate a line, what we're going to basically be asking is, what's the slope that reduces this sum squares value, the sum of the squared residuals across the data set. Numerator of the sample variance, super important. If you understand this, you understand so many things about doing different types of statistics. This is what we used in ANOVA. This is also what we're now using in regression. So the mechanics by which you do this is kind of complicated. And if you take an applied stats class, which I hope you do, when you're further along at William Jewell College, I will teach you actually the mechanics of estimating the slope and things like that. But basically, you could do this in R, you could do this in Excel, wherever you want to do it. But basically, what regression analysis does is there's an equation that you can use to calculate the slope. And when you get the correct slope, here's the equation. The slope value is negative 0.0532. The y-intercept is 8.704, which you see is right up there. And what it has done is it has reduced overall levels of residual variation. All of these data points are now much closer to the line than they were before. And so you can think of this line as the running average of weight loss as weight loss declines with increasing relative humidity. So one of the ways of thinking about the regression line is the average. But it's the average value of weight loss at a particular value of relative humidity. It's the average of Y at a particular value of X. But because we can think of it as an average, we can use things like the sums of squared deviations from the mean to evaluate the model. So now our residuals are all really tiny. So there's this other thing that you can get printed out, R-square. R-square has to do with how variable the data are around the line. In this case, the data are not very variable around the line, and R-square is 0.9. 0.97 rather. R-square can vary from 0 to 1. 0 is when the data are so spread out that well, let me explain what R-square means. The easiest way to think about R-square for me is what amount of variation in the Y variable does the X variable explain? What's another way of saying that? What proportion of variation in Y is explained by variation in X? So in this case here, we have a linear relationship where Y equals X, and every point is along the line. So the slope is 1 and the y-intercept is 0. So this is also essentially the same as saying y equals 1x plus 0, where 1 is the slope, 0 is the y-intercept. Well, that just basically reduces down to y equals x. So in this case, if you know x, what do you know about y? Not a rhetorical question. If you know x, what do you know about y?
As x increases, y increases.
Oh, you know more than that. What do you know about y?
X is Y.
You know everything there is to know about y. Y is totally transparent to you because variation in x explains all of the variation in y. And so r square is 1.00 because variation in x explains 100% of the variation in y. If the data are a little variable around the line, does x still tell you something about y? Joseph, you're nodding your head, yes? Speak. That was a command, Joseph. You're supposed to speak. Does X tell you something about Y?
Yes.
Does it tell you quite a lot about Y?
Tells you a good amount, I think.
Yeah, it tells you, so 90% of the variation in Y is explained by variation in X. So X doesn't tell you as much about Y as in this case, but it's still telling you quite a lot about Y. If you want to know what Y is and you're in the region of 60, you have a pretty good idea of what Y is going to be, but not as good of an idea as you did under this scenario. As those data get more spread out, what happens to your R-square value? It goes down. So now only 69% of the variation in Y is explained by variation in X. If you know something about X, do you still know something about Y? Yeah, you do, but it's less firm than it was before. And we can continue doing this until finally we have data that are so spread out that the R-square value becomes almost 0. So this is from an actual paper published in the Journal of Evolution back in the 1990s. And you can see down here in the figure caption that the R-square value here is 0.09 for this plot, and the R-square value is 0.075 for this plot. So R-square of 0.075 0.09, and 0.075. In this case, only 9% of the variation in y is explained by variation in x, and only 7.5% of the variation in y is explained by variation in x. Now, also when you look in this figure caption, you'll see that these are highly significant regressions. We know that that slope is different from 0, and we know that that slope is different from 0, but part of that is that there's a huge sample size here. There's 338 different bird pairs that are in this plot. There are 338 bird pairs in this plot. And so the regression is significant. You can tell that this line is a line different from 0 because you've killed it with sample size. But male tail length doesn't really explain much of the variation in female tail length. And this is one of those weird things in statistics where statistics, in this case, having too many samples is actually kind of a bad thing because it makes you think that this line is much more significant than it actually is, because as you increase sample size, your p-value goes way down. But the explanatory power of male tail length is not very good at explaining female tail length. So you have a significant regression that has a really low explanatory power based on R square. What was I about to say? A significant regression that doesn't really explain very much of the variation that you're interested in doing. And so it's probably not as biologically relevant as it could be, simply because knowing male tail length doesn't tell you a lot about knowing female tail length. So R square is important, but that's not what the regression is testing. The regression is testing whether or not the slope is different from a slope of 0. Okay, so make that distinction in your head. This test that has a p of 0.0001, that's testing whether or not the slope is different from the slope of 0. R square tells you how useful is male tail length at telling us about female tail length. Is that clear? Okay. We haven't done this yet, but we'll do this on Friday. We also use regression to estimate heritability when we look at parent-offspring regressions. Just another application of it. I was going to do the shoe size data set with you in class today, but I decided not to. We're just going to actually go through the analysis with the beetle data. But I do want to show you some of the reasons that this is important. Having this ability to do this is you could ask whether men and women have different shoe sizes. You would typically examine this using an ANOVA, but unfortunately, when you look at this, you find that males have larger shoe sizes than females. But the reason for this is actually that shoe size co-varies with height. And because males are generally taller than females, we should expect them to have larger shoe sizes than females. This is the data without jittering it. In R, you can jitter the data so you can see the data points, but you can see that most of the male data is up here and most of the female data is down here. And so the difference in shoe size is actually just differences in overall height that is separated by gender. But this line is fairly consistent in terms of describing the relationship between shoe size and height. Here's the slope value. There's the y-intercept. There's 408 values in this data set. The F statistic is ginormous. Once again, because we have 408 observations, it's going to produce a really tiny piece, statistic, 2.2 times 10 to the minus 16th, and the R square value is 0.758. So basically, about 76% of the variation in shoe size is explained by variation in height, is what that's telling you. The slope is very significantly different from 0, 0.42, and the p-value tells you that it's not anywhere near 0. So that's kind of the basic concept behind regression. questions about that? Alexis is smiling nervously like, I want to ask a question, but I don't want to ask a question.
I mean, I think I'm fine.
Okay, all right, well, good. If everybody's fine, let's go to R then. So in R, let's go back to the Bio 133 lab stats folder. I have a regression example here worked out for you, and it's going to be based on the beetle data set. So in R, you can clean up your workspace by using the little brooms here. So you saw that I just got rid of all the plots from the ANOVA thing. And so here I have the regression analysis, and go up to the top. The first thing you will always want to do is check and make sure that you're in the right place. I am in Bio 133 lab stats. I'm going to add the packages that I need. I'm going to download the Beetle file. I'm going to attach the Beetle file. And because the Beetle file is so small, I can just ask it to give me the Beetle file. And there are the beetles. And that should look just like what it looked like before. And so as humidity goes down, weight loss goes up. So I have the Beetle file available to me. I know that it looks like that. So in linear regression, we specify the model a little bit differently, but it's not that different. We're going to use linear models now instead of analysis of variance, but the syntax is kind of the same. So I'm going to create a shorthand thing here called model 1, and I'm basically asking it to build a linear model where weight loss, whoops, I don't want to move things, where weight loss is influenced by relative humidity, and I want you to do this on the data file, the data frame beetles. So whenever you see, so in this case, it's linear model, but we could have done an analysis variance as well. So whenever you see weight loss tilde humidity, this is the dependent variable and this is the independent variable. And that applies whether you're doing an analysis of variance or whether you're doing a regression. So in the case of the plants, growth tilde treatment, it's basically saying, what is the effect of treatment on growth? What is the effect of relative humidity on weight loss? And so that's the syntax there. So this is just going to run the regression model. Hopefully it runs it without error. There are no error messages. So once again, we can also get a summary of this regression model. And in the summary of the regression model, it's going to give us the y-intercept, which should match the y-intercept from the PowerPoint. And it gives us the slope value, which is negative 0.053222, which is also the slope that was in the PowerPoint. And it gives you the p-values. And the p-value that you are interested in here is the p-value for the slope, because that's what a regression tests. It's testing whether the slope is different from 0. Now, this slope is not very far from 0. It's a really shallow slope, but it's a significant slope in that we can distinguish it from a slope of 0. Here's the R-square value, 0.9745, which is what we looked at earlier. This is the F statistic, and you have 1 in 7 degrees of freedom because you have 9 values, and there, once again, is the p-value, and it's the same as that p-value up there. You can also have this produced in an ANOVA table. So we can look at the ANOVA table. It has sums of squares due to the regression model, and sums of squares of this residual variation. You see that the regression model explains a lot of the variation in the data, and there's very little residual variation, which is why this, this F value ends up being ginormous. And so we can figure out the total sums of squares just by adding the sums of squares due to the regression to the sums of squares due to residual. And if we divide the sums of squares due to regression by the total sums of squares, that gives us the R-square value, 0.9744716, which is 0.9745, essentially. So the calculation for what R-square is basically how much of the total variation in Y is explained by the regression line. And so to what degree does variation in X explain variation in Y? That's what we're calculating when we calculate R-square. How much of the sums of squares is due to regression out of the total sums of squares that we have? Okay, so that's actually pretty important. So we can calculate what the relative humidity mean is. It's 50.3. We can calculate what the weight loss mean is. It's 6.022. We can find out what the residuals are. So here we see the residual variation. It's really high at low relative humidities, and it's really high at high relative humidities. And in the middle of this range, it's close to 0. Well, this is when we just compare it to the mean weight loss, but we can also get the residuals from the model. So we have model 1 here, which is just the regression model. Well, we can get R to tell us what the residuals are, and we can tell it to store it in a new column in the Beatles data file. So up here, we told it to take the residuals from the mean and put it in a new column in the data file, which is why we got WLRes here. But we can do the same thing again and look at the differences in the residuals. So this is the weight loss residuals when we were just comparing the values of weight loss to the means right here. This is the residuals as a result of refitting the line to the correct regression line. And you can see how much smaller these residual values are than these residual values. And that's exactly what regression is supposed to be doing for us. So once again, this is going to be a plot. In this case, the aesthetics are humidity and weight loss. We have Beatles as the data frame. We're going to use a smoothing method. The smoothing method is a linear method. That line is going to be black. It will also add 95% confidence intervals around the slope of the line. We're going to also tell it to give us some points. We're going to label our things. We're going to give it a title. We're going to format the thing. When we run all of this, it should just give us a graph. And the graph should look exactly like the graph that we had before. And once again, this is the regression line. The residual values are really small around that regression line. The gray band is the 95% confidence interval of the slope. So to visually evaluate a regression, you could say the average value is, let me go back up here. I have some things in here that I had commented out. I'm going to rerun this thing. The average value of humidity is just over 50%. The average value of mass loss is just over 6. This makes a fulcrum right here in the middle of the data set. If we tilt that line on that fulcrum to a horizontal line, will that horizontal line fit within that gray band? If it will, then your slope is not different from a slope of 0. If it won't, your slope is different from a slope of 0. And you can see that if we tilt that line at that fulcrum, it doesn't take much tilting down towards a slope of 0 to take that line outside of that gray band. And that's another way of kind of visually ascertaining whether or not your regression slope is different from a slope of 0, in the same way that we can look at means and 95% confidence intervals in an ANOVA to evaluate that visually. You can also evaluate that visually with a regression. Does that make sense? You guys feel okay with this? Better than just watching the screencast? Probably do more freestyle riffing in this venue than on a screencast. Okay, you just got like 5 weeks of applied statistics in under 2 hours. So if you have to do regression as part of your project, we will help you with the regression. If you have to do ANOVA as part of your project, we will help you with that. We're not expecting you to be able to just whip this stuff out right now. So don't put that pressure on you. We are here to help you through this process, but we want you to be familiar enough that you can interpret these things once you do see them. And when you're playing around with it with your own data, it will begin to make a lot more sense than these stupid examples that I pulled out of a textbook. All right, other questions? All right, Dr. Judd and I are working feverishly to get the protocols written. Once we have the protocols written, we will get them up on Moodle. The protocols are written in kind of step-by-step fashion. In many cases, they have photographs of the steps. And in some cases, we're actually going to be putting videos of us doing the methods on the YouTube site. So when in the protocol it says refer to YouTube for that, go to the YouTube site and you'll see a video of, for example, me in the field with an insect net collecting stinging insects versus unstinging insects. If you're collecting beetles, if you're in the pollen load group, if you're collecting beetles, you can just hand collect those right off of the goldenrod. If you're collecting wasps and bees, you need to collect those in an insect net and kill them before you get them out of the insect net. And so I went out, like I said, I went out yesterday and shot a video that I need to now edit together that basically kind of takes you through that process. When you go out and collect, like I've said before, if you need Dr. Judd or I to go out and assist you with that, by all means, make an appointment with us to do this stuff. Especially if you're working with insects, you probably want to schedule your field work in the middle of the day when it's warmer. So it was warm and now it's much cooler. I was hard pressed yesterday to find a wasp to collect to make a video because I went out at the end of the day because I had classes all day, then I had research meeting, and so I didn't get out there until like 5:30. And quite frankly, I could have collected that wasp with my fingers without any danger, but I collected it in a net because that's how I want you to do it. But it was just too cold for things to be moving very well. Hopefully we'll get some warm weather before it then cools off permanently. But for insect work in particular, it's going to be important that you be out there in the warmest part of the day because that's when you're going to have the most activity. So I'll send out an email when all of the protocols are posted, but keep an eye out on Moodle as well. And if you have Moodle notifications on, then it'll show you when those things were loaded up, more than likely. Once those protocols are up, you need to read through them, and then you need to schedule time with your group members if there's field collecting that you need to do as a member of a group, or if you're doing individual work, schedule it into your week just like you would anything else to go and get it done. And we'll have instructions on where supplies are in the lab. And as always, if you have any questions about what you need to be doing or how you need to be doing it, let us know. We would rather clarify what you're doing before you go out and waste a bunch of time doing something incorrectly in the field. We would just rather, make sure that you know how to do it before you go out so that you're not wasting your time in doing this. For social distancing, I would prefer that you work, individually when you can. Now, that doesn't mean that you have to go out by yourself. It just means that if you're out with your other team members, that you do your thing over here and somebody else do the same thing over there, some distance away. If you're appropriately distanced outside, if you're out there with your whole team and you're doing individual collections, but you rode there together, masked up in your car, that's fine. And if you're 15, 20 feet away from one another, you're collecting on this on this patch of plants over here, and somebody else is collecting on that patch of plants over there. There's no reason for you to be masked if you're uncomfortable in the field being masked, if you're sufficiently distant from one another. So you can schedule your field work together as a team. Just the closer you are out in the field, the more necessary it is for you to be masked up. If you're a sufficient distance away, then you can just relax and breathe in the fresh air. Questions? All right, that's all we have for you today. So look for the protocols to appear.
I have a quick question, sorry.
Yeah, Joseph, what's up?
Do we need to submit the data management and goldenrod's assignments anywhere?
Yeah, normally we would meet in class, so you can just drop those because they're handwritten on paper. Just drop them by my office.
Okay, cool.
Yep, that'll be fine.
I have a question. We have a assignment about the goldenrod. It's like the getting to know goldenrod. Do we email that
Yes.
to you?
Yeah, you can either email it to me, or since it's on a piece of paper, once again, you can just drop it by. So both of the assignments that were for today, either email them to me, which a couple of you already have, or just drop the hard copies by my office.
Okay.
Actually, I prefer hard copies. So one of the reasons I didn't get protocols finished yesterday was the act of moving around all of the digital files that were associated with your exam and making sure that I didn't accidentally send one person's exam to another person, which would be a violation of FERPA. That took way longer than I expected it to. I basically did two things. I taught class, I went to a research meeting, and I dealt with your exams yesterday. So I myself am growing tired of digital learning because manipulating files is just a royal pain in the keister. So yeah, if you want to just drop them by handwritten, I will be happy to take them from you. And I'll grade them handwritten and I'll hand them back to you handwritten. Yeah,
I
Paula.
already wrote down everything on my iPad, so I'm just gonna print it out. Is it okay?
No, you don't have to print it out. You can just send that to me email. That's fine.
Okay.
Save your printing allocation for something else. Like I'll deal with digital files. I just found myself a little overwhelmed with the exam. But now I've hopefully we've developed some systems after the first one. Maybe the next exam won't be quite so grueling.
So you said for the like not the getting to know you one, but the other assignment that we only needed one of those turned in per group for like the
Yeah, so your data management worksheet you worked on as a group. So yeah, I just need one of those from each group, but I need to have all the group members' names on it. Yep.
Perfect.
Yeah. All right, that's it for today. I'll see you tomorrow in class, and I'll see Tessa shortly. You still coming over?
I was gonna email you about that. I have a class during your office hours.
Oh, well, why don't you just come over now?
Okay, I can do that.
Yeah. Okay, all right. All right, I'll see you guys tomorrow.
I actually had one
Yeah.
more question. So
Yeah, Alex.
the getting to know goldenrod, that's a personal assignment. So we
Yes,
each
that's
are
an
doing-
individual assignment. So sometime between last week and today, you should have gone out and looked at some goldenrod in the field.
Oh yeah. Okay.
Yeah. Yep.
Sounds good. Thanks.
Okay. All right.